We have already completed structure of microspore and structure of megaspore along with microsporogenesis and megasporogenesis. Today's session continue with the heading pollination. You know that in sexual reproduction there are three phases: pre-fertilization, fertilization and post-fertilization. In pre-fertilization there are two steps: gametogenesis, that is production of gametes, and the transfer of gametes, followed by fertilization, that is fusion of male and female gametes to form zygote. And this zygote undergo cell division, cell differentiation, which are the two events in embryogenesis which occur under post-fertilization event. So in this topic, sexual reproduction in flowering plants, gametogenesis refers to microsporogenesis and megasporogenesis, which give rise a mature microspore or pollen grain. It is known as male gametophyte, and a mature megaspore or ovule in the ovary. Now, as both are non-motile, microspore and megaspore, so they have to brought together for fertilization. So this event of transfer of gametes in flowering plants is known as pollination. So let us begin with the definition. The transfer of microspores or pollen grains from anther to the stigma is known as pollination. This process is classified into two types depending upon the source of pollen grains and its transfer. Self-pollination, which occur in a single flower; cross-pollination occur in two flowers, either growing on the same plant or growing on different plants belongs to same species. So in today's session, I will complete first type of pollination, that is self-pollination. It is known as autogamy. The transfer of microspore or pollen grain from anther to the stigma of same flower is known as self-pollination or autogamy. Now, what are the advantages of self-pollination? Number one, it ensures 100% pollination. Number two, it maintains purity of the species. It is also known as it maintains homozygosity. The number of microspores generated and number of megaspores produced will ensure 100% rate of fertilization. But continuous self-pollination or autogamy leads to inbreeding depression. It is a disadvantage of self-pollination or autogamy. Now, what is inbreeding depression? After 5-6 generation, if the self-pollination continues, then it results into loss of fertility and loss of productivity. So, inbreeding depression is a disadvantage of self-pollination or autogamy. Now, when this self-pollination is possible, there are contrivances or there are some conditions. For self-pollination, flower should be bisexual with androecium and gynoecium. This is first condition. Second, they should show homogamy, means microsporogenesis in anther, megasporogenesis in ovary should occur at the same time, followed by maturation of microspore and maturation of megaspore, which takes place at the same time. So both remain fertile. This we call homogamy. There is a third condition which is rarely found in the flowers of pansy or viola. So in viola or pansy flowers, some flowers are clistogamous, some flowers are chasmogamous. Clistogamous flowers do not open, they never bloom. They are completely in closed condition. While in chasmogamous flowers, the flower opens. So here in pansy or viola, the flowers which are clistogamous, which is favoring self-pollination. Chasmogamous flowers favors cross-pollination. So which are the three conditions for self-pollination or autogamy? Number one, the flower should be bisexual. Number two, the flower should show homogamous condition. Number three, the flowers should be clistogamous. They do not open, they are permanently closed. The question will appear on this part is definition of pollination, types of pollination, under autogamy advantage and disadvantage, what is inbreeding depression, and what are the conditions or contrivances for self-pollination or autogamy. Hope it is clear with self-pollination. I'll continue with the next type, it is allogamy or cross-pollination. Now in allogamy or cross-pollination there are two types, geitonogamy and xenogamy. In geitonogamy, though it mimics cross-pollination, but it maintains homozygosity because here the transfer of microspores or pollen grains occur from anther to the stigma of another flower growing on the same plant. So plant is same but two flowers are involved in pollination. Okay, if a twig of the plant having two flowers, from A flower the microspores are dropped on the stigma of B flower. Now as I said, when the two flowers are involved in the process, it is a cross pollination, but it will not give variations. It maintains the purity of the species. Okay, only by transferring the pollen grains from one flower to another flower, it mimicking the cross pollination or allogamy. Got it? Now, the next type of cross pollination is called xenogamy. It always brings out variations. Here, the microspores are transferred from anther to the stigma of another flower growing on different plant, but both plants belongs to same species. For cross pollination or allogamy, to transfer microspores, okay, they require agents. Now there are two types of agents, abiotic and biotic. Abiotic agents are non-living agents. For example, water and wind. Biotic agents are the animals. Now in animals, maximum insects will pollinate the flowers. Some flowers pollinated by the birds. Some flowers which blooms in the night pollinated by the bats. And some flowers are pollinated by the snails. So all these animals are called pollinating agents. So cross pollination require abiotic agents like wind and water. Biotic agents are the animals. Okay, that we will study in the next session. Here in cross pollination, what are the conditions required? Number one, cross pollination occur when the flower is unisexual. Means staminate flowers will produce the microspores and pistillate flowers produce the megaspores. So here there is no option. From one flower microspores are transferred to the stigma of another flower. This is one condition known as unisexuality. Second condition, flower is bisexual with androecium and gynoecium, but they show dichogamy. In dichogamy, first androecium matures, means microspores will mature early than the megaspore. This we call protandry. There are some flowers show reverse condition, means first megaspore develop and then microspore. This we call protogyny. So under dichogamy condition, maturation time differs. Based on that, we call it as protandry. Androecium matures first and protogyny gynoecium matures first. Third condition, if the flower is bisexual but there is a barrier, this barrier is not allowing the deposition of their own pollen grains on their own stigma. This condition we call it as hercogamy. Here, either the filament of the anther is long, known as heteroanthy. There is another condition, length of the style is maximum, which will take the stigma outside the flower, which will not allow the deposition of their own pollen grains. This we call heterostyly. So heteroanthy, heterostyly together known as hercogamy. So what are the conditions for cross-pollination? Unisexual flowers, bisexual flowers showing dichogamous condition, either protandry or protogyny. Hercogamous flowers either showing heteroanthy or heterostyly. Fourth condition is self-sterility. Means if pollen grains deposits on the stigma of the same flower, then they are unable to germinate, or stigma will not allow pollen grain to germinate. This self-sterility or incompatibility which brings about cross-pollination because it accepts the pollen grains of other flower. Okay, even if their own pollen grain deposit, they won't allow. So let us list the conditions of self-pollination, conditions of cross-pollination. For self-pollination, flower should be bisexual, homogamous condition or clistogamous condition. For cross-pollination, unisexuality, dichogamy, hercogamy, self-sterility or incompatibility. With this, I will end today's session. Next, about the pollinating agents, we will study in the next topic. Thank you.